welcome to The Explainer. Let's get right into it. If you're a brewer, a food technologist, or a bioprocess engineer, you already know that fermentation is the absolute heart of the brewing process. It's this incredible intersection where yeast physiology, metabolic biochemistry, and process engineering all collide to dictate the exact quality of your final product. Today, we're taking a deep, literature-grounded dive into how these single-celled eukaryotic factories actually operate. Because let's face it, understanding exactly what's happening at the microscopic level is the only real way you get macroscopic control over your product.
To break this all down, here's our game plan for today. We'll hit 1. Yeast taxonomy and structure, 2. Fermentation biochemistry, 3. Flavor compound generation, 4. Fermentation growth kinetics, and wrap up with 5. Key process control parameters.
All right, starting off with Section 1, Yeast taxonomy and cell structure.
So, right out of the gate, we have to look at the genetic and operational split between our two main brewing yeasts. On the left, we've got Saccharomyces cerevisiae, our classic ale yeast. It's a top cropper. Why? Because CO2 bubbles literally stick to its cell walls and float it right up to the surface. It loves warmer temperatures, maxing out around 37 degrees Celsius, but it totally lacks the ability to ferment the sugar melobiose. Now look at the right, Saccharomyces pastorianus, the bottom-cropping lager yeast. The really cool thing here is that S. pastorianus is actually a natural hybrid of S. cerevisiae and a cold-tolerant wine yeast called S. eubayanus. Because of a different cell wall structure, it sinks. It demands cooler temps with a strict max of 34 degrees Celsius, and unlike ale yeast, it absolutely can ferment melobiose.
And when we zoom in to map out the subcellular machinery, it's like looking at a highly specialized factory. You've got the rigid cell wall, and just inside that is the plasmalemma, or cell membrane, acting like the bouncer, regulating exactly what wort nutrients get inside. See those little rings on the outer edge? Those are bud scars. They're literally physical markers of cellular age. The more scars you see, the older that parent cell is. Inside the cytoplasm, you'll spot the vacuole, a storage organelle that actually swells up as the cell gets older. And you've got to pay attention to the mitochondria. Now, they aren't churning out energy via aerobic respiration during fermentation, but they are absolutely essential here. Without them, the yeast simply cannot synthesize crucial lipid compounds, specifically fatty acids and sterols.
Moving right along to Section 2, Fermentation Biochemistry and Theory.
Okay, so here is the core biochemical reality. When yeast respires aerobically with oxygen, it completely burns sugars into CO2, water, and a ton of energy. But under anaerobic conditions, which is exactly what fermentation is, this process gets incredibly inefficient. Glucose is just partially broken down into alcohol, carbon dioxide, and way less energy. For the yeast, it's basically a desperate survival mechanism to keep the lights on without oxygen. But for us, well, it's the precise mechanism that gives us that sweet, sweet ethanol.
This physiological shift is just brilliant. Depending on the environment, the yeast completely flips its behavior. Under aerobic conditions, it uses oxygen and fermentable sugars to generate the energy needed for massive, rapid cell reproduction. It's basically in full population boom mode. But strip away that oxygen, and cell growth practically hits a brick wall. Instead, under anaerobic conditions, the yeast transforms into a conversion engine. It starts furiously turning those sugars into alcohol, CO2, and a whole cocktail of aromatic flavor compounds.
And to keep that conversion engine running, yeast needs a lot more than just sugar. It needs a very specific diet. Amino acids pulled from barley protein during mashing, lipids, vitamins, and trace elements like calcium, zinc, and copper. When the cell processes all of this anaerobically, it doesn't just neatly output ethanol and CO2. It actually leaks a highly complex matrix of metabolic byproducts. We're talking higher alcohols, diacetyl, acetaldehyde, sulfur compounds, and esters. And honestly, these biological leaks, they are the absolute key drivers of your product's entire sensory profile.
Which brings us perfectly to Section 3, Flavor Compound Generation.
So how do we actually control those leaks? Well, specific process variables act as the physical levers for different flavor classes. Let's look at esters. These give you those great fruity tropical notes, like the banana bomb of isoamyl acetate or the boiled sweet vibe of ethyl acetate. Esters are created when fatty acids bond with alcohols. The key lever here is the ratio of fermentable sugars to assimilatable nitrogen, or FAN. Push your wort gravity higher and you're gonna drive up your ester yield. Higher alcohols, on the other hand, give a harsher wine-like character, and they are directly tied to yeast growth. If you boost wort oxygen, raise the temperature, or increase fan, you turbocharge yeast growth, which inevitably cranks up higher alcohol production.
Now let's talk about diacetyl. This is the compound that tastes like butterscotch or toffee. Sure, it's sometimes acceptable in certain traditional ales, but in a crisp lager, it is a massive, severe flaw. If you're brewing lager, you have to drive this level down below a really strict sensory threshold of just 25 parts per billion. Diacetyl gets synthesized during active fermentation, but the magic trick is that the yeast will actually reabsorb it and reduce it into much less flavorful compounds, provided you give it a warm conditioning phase. You literally cannot chill that tank down until your lab confirms you've crossed that 25 ppb finish line.
Similarly, we have dimethyl sulfide, or DMS, which has a sensory threshold of 30 parts per billion. Hit that threshold, and your beer is going to taste dead-on like cooked cabbage or tinned sweet corn. But unlike diacetyl, DMS is primarily a malt problem, not a yeast problem. You tackle this upstream. You control it by demanding strict malt specifications from your maltster, and then by hitting your wort with a vigorous rolling boil to literally volatile the DMS before it ever sees a yeast cell. Other sulfur compounds, like hydrogen sulfide, that nasty rotten egg smell, those rely on a super vigorous fermentation so the escaping CO2 can just scrub them right out of the liquid.
All right, let's look at the life cycle in Section 4, Fermentation Growth Kinetics.
To really grasp what's happening over a standard 7-day fermentation cycle, we have to track 3 things: specific gravity, yeast count, and temperature. Take a look at the gravity curve. Notice how it doesn't just instantly drop. It actually flatlines for a bit, while the yeast population is quietly building its strength. But then, boom, as the yeast count goes exponential, the specific gravity absolutely plummets. It's a perfect visual representation of heavy sugars being converted into lighter ethanol and escaping CO2. If we break that timeline down, we see 4 distinct biochemical phases. First is the lag phase. Gravity is totally stable here, while the yeast acclimatizes, soaking up oxygen and nutrients like a sponge. Next up is the log, or exponential phase, kicking in about 6 to 12 hours after pitching. This is explosive. Cell numbers can double every 90 to 120 minutes, and this is exactly when your gravity drops off a cliff. Following that is the retardation phase. Growth finally hits the brakes because fermentable sugars and FAN are running dry, while toxic alcohol levels are climbing. And finally, the stationary phase. The death rate is now outpacing the birth rate. But don't rush this. This phase is absolutely vital because it's when the surviving yeast acts like a cleanup crew, mopping up leftover nutrients and reabsorbing that diacetyl we mentioned earlier.
And that brings us to Section 5, Key Process Control Parameters.
Let's start with wort oxygenation. Now I know what you're thinking. Fermentation is strictly an anaerobic process, right? So pumping oxygen into the wort sounds completely counterintuitive. But here's the catch. The yeast doesn't use this oxygen for cellular respiration. It absolutely requires it to synthesize storols and unsaturated fatty acids. These specific lipids are non-negotiable for building strong cell membranes that can actually survive the toxic stress of an anaerobic high-alcohol environment. If you miss the mark on oxygen dosing, you're looking at sluggish fermentations, under-attenuation, and extremely weak yeast that you definitely cannot repitch.
Next up is your pitching rate, the exact volume of cells you inoculate the wort with. There's a massive difference in the science here depending on your strain. For standard ales, you're looking at roughly 9 million cells per milliliter. But if you're bottom cropping with a lager yeast, you need way more firepower. We're talking 14 million cells per milliliter at a minimum, and even higher if you're pushing into high-gravity territory. You literally have to brute force the cell count to guarantee a healthy, vigorous fermentation at those highly suppressive cold operating temperatures.
So when we pull back, we see that these specific, actionable controls are the real-world levers process engineers use day in and day out. Pitching rate and dissolved oxygen, that dictates your baseline yeast health. Your temperature profile, meaning the initial pitch temp, how fast it rises, the top heat, and your final crash temp, that acts as the main throttle for both growth speed and flavor synthesis. But actually, one of the most fascinating variables isn't chemical at all. It's the physical geometry of your tanks.
Which brings us to a final, pretty provocative engineering question to leave you with. Most modern facilities use these massive, towering, cylindro-conical fermentation vessels. The sheer volume and weight of the liquid in these things creates immense hydrostatic pressure down at the cone. And here's the kicker. That intense pressure physically suppresses yeast growth.
And as we established earlier, if you suppress yeast growth, you actively shut down the synthesis of both esters and higher alcohols. So looking at your own operations, how do you balance the sheer physical reality of that hydrostatic pressure with the incredibly precise, targeted flavor profile you're trying to coax out of your yeast? It's a massive puzzle, and honestly, that's the true art and science of fermentation engineering. Thanks for joining me on this explainer, and keep digging into the science behind your process.